For decades, the medical community has viewed cannabis and cancer care through a very narrow lens. It was prescribed as a palliative tool, a way to help patients manage the severe pain, chronic nausea, and loss of appetite that inevitably come with conventional therapies. Standard treatments like chemotherapy and radiation are intentionally harsh. They rely on blunt toxicity to kill rapidly dividing cells, which ravages the body and often leads to a secondary hurdle. The surviving tumor cells mutate and develop drug resistance. These laboratory results show a different story. This photograph from a preclinical study compares excised tumors from two groups. The top row is the control group. The bottom row shows tumors from the group treated with cannabidiol. The extract is actively shrinking the tumors. This physical reduction happens because cannabinoids interact directly with the body's endocannabinoid system, a complex network of internal receptors that regulate cellular function, including how cancer cells survive and proliferate. We are left to investigate how a non-intoxicating plant compound systematically hunts down and destroys aggressive cancer cells while leaving surrounding healthy tissue completely unharmed. Normal biological cells have a built-in expiration date. When they become severely damaged or too old, they receive a chemical signal to destroy themselves. Cancer cells survive by ignoring this signal, rendering themselves biologically immortal. CBD overrides this glitch. Breaching the cell membrane, it forces the production of highly volatile reactive oxygen species, or ROS. These flood the cell, driving internal pressure to lethal levels. This overwhelming stress trips a biochemical wire called apoptosis, initiating a highly organized self-destruct sequence. This targeted mechanism turns the cancer cell's corrupted software against itself, forcing the tumor to execute its own destruction. To grow beyond the size of a microscopic cluster, a tumor needs a continuous supply of nutrients and oxygen. It gets this by hijacking the body's infrastructure, forcing it to build a dedicated network of new blood vessels, a process called angiogenesis. CBD chemically intercepts the signals the tumor uses to demand those blood vessels. By downregulating specific pro-angiogenic factors, it halts the construction of new supply lines, actively starving the tumor mass. This chart illustrates the downstream effect of cutting off that fuel supply. Looking at chart C, we see a stark reduction in the number of metastatic lung nodules in CBD-treated groups. Without a robust network, cancer cells cannot easily migrate and invade other organs. The extract functions as a dual biological blockade. It cuts off the incoming fuel supply and traps the existing disease in place so it cannot spread. The human immune system is highly efficient at clearing out cellular threats, provided it can actually see them. Cancer cells survive by deploying molecular camouflage, cloaking themselves to glide past patrolling white blood cells unnoticed. CBD strips away this disguise, forcing the tumor to express surface proteins called ICAM1. These act as bright, high-visibility flags permanently attached to the cancer cell. Specialized immune responders called LAK cells finally recognize the threat, locking onto these markers to dismantle the exposed cell. By forcing the tumor to reveal its location, the compound acts like a biological flare gun, weaponizing the patient's own immune system to clear the disease. Despite these mechanisms, clinical oncologists rarely view CBD as a standalone cure. Its most powerful application in modern medicine is as an adjuvant, a therapy used directly alongside traditional chemotherapy. Over time, cancer cells mutate to survive the toxic flood of chemotherapy. They build specialized protein pumps on their outer membranes that catch the toxic drugs and spit them back out before they can do any damage. CBD binds directly to these resistance proteins, such as PGIP. It physically jams the cellular pumps, trapping the chemotherapy inside the cancer cell where it belongs. By disabling the tumor's primary defense mechanism, it operates as a biochemical amplifier, making standard chemotherapy lethal to cells that had previously figured out how to survive it. These microscopic interactions are now making their way into human trials. The laboratory data is beginning to translate into observable, documented clinical outcomes for living patients. Peer-reviewed case reports detail prolonged survival periods in patients with highly aggressive brain tumors, like glioblastoma. Verified tumor regressions have been documented across forms of leukemia, prostate cancer, and breast cancer when treated with exact ratios of these cannabinoids. There is, however, a significant bottleneck. Standardizing this treatment requires massive, randomized human trials to pinpoint precise dosing protocols across different cancer types, which are currently being pursued. The path toward clinical adoption is complicated by the immense cost of phase 3 trials and the regulatory hurdles inherent to standardized plant-derived medicines. These factors often slow the transition from the lab to the pharmacy. The clinical data confirms CBD operates as a highly targeted, multi-pathway biochemical tool. It strips away drug resistance, starves tumors of their blood supply, and forces them to destroy themselves, marking it as a vital asset in the future of integrative oncology.